Well, welcome everyone. My name is Christine Santora. I work here at Stony Brook University with the Shinnecock Bay Restoration Program, and I've worked with Ellen for over 20 years, and I'll be introducing her. So Dr. Ellen Pikitch is the founder and executive director of the Institute for Ocean Conservation Science at Stony Brook University. She's also an endowed professor of ocean conservation science. Ellen's contributions have resulted in scientific innovations, fisheries management improvements, and domestic and international policy change. Ellen is frequently called upon to provide her expertise at professional meetings, global scale fora, and to decision makers at the state, national, and global levels. Ellen focuses on topics like marine protected areas, the use of eDNA technology to assess biodiversity, fisheries management, and protecting endangered species. She's worked at the United Nations to bring the science of MPAs to countries around the world and to encourage nations to achieve their goals of protecting, at the time, 10% of the ocean by 2020. Ellen is an active fellow of the Explorers Club and recently won its Lowell Thomas Award in 2024. She's won other prestigious awards, including Mission Blue's Hope Spot Champion Award, the Oscar Sett Award from the American Fisheries Society, and the Ocean Hero Award. So I don't know how many of you know, but Ellen is a native New Yorker growing up in the Coney Island area. But she went to the City College of New York to earn her bachelor's and master's degree in mathematics. She went on to Indiana University to earn her master and doctoral degrees, and then she went on to hold academic positions at the University of Oregon and the University of Washington. And I will add, being a pioneer woman in academia at that time, especially doing fisheries research. So she moved back to New York in the late 90s to found and grow the marine program at the Wildlife Conservation Society. And any of you who are familiar with WCS, they have many on-the-ground conservation programs in many different countries. And Ellen found there was no marine program at the time that Ellen went to WCS, and she founded a global marine program. And she impacted on-the-ground conservation efforts in 18 countries in 4 oceans. She co-founded the Shinnecock Bay Restoration Program with Chris Gobler in 2011, and you will hear much more about her fisheries research tonight. So I've worked with Ellen for over 20 years, and she really is an excellent, not just scientist, but visionary and communicator. She's passionate about cutting edge science and using that science to apply it to make a difference in policy change and to make a difference in people's perception of the oceans and why they're important. So thank you, Ellen, for giving this talk tonight. And we're all proud to be part of your team. And I'll hand it off to you.
Thank you, Christine. Were you really talking about me? That was an amazing introduction. Okay, so Christine has told you that, I've worked at the big global level, and here I am working on a very local issue, Shinnecock Bay. How many of you have stood on the shore of Shinnecock Bay? Okay, how many of you have been on the water of Shinnecock Bay? Wow, this is a very experienced audience. How many of you have gone swimming or snorkeling or scuba diving in Shinnecock Bay? Okay, so not everyone. All right, so some of this will be, that's good to know. So we have a very educated audience, experienced audience in Shinnecock Bay. I'm going to give you an overview of what I and others have been doing for more than a decade in Shinnecock Bay. So what got me interested in Shinnecock Bay? In 2008, I went from the Wildlife Conservation Society to Stony Brook University. And I decided, it would really be good to have a local project, something where students and I could get out to frequently. And that was really my motivation. I was looking for a place that was close. Stony Brook, as you are in here, has a marine station, a fleet of research vessels, what better place to start a research program than right here? I also was looking for a place that had problems, because one of the things I like to do is fix problems. I love a good problem, and when I hear something's in trouble, I say, sign me up, I want to try to fix it. And boy, did Shinnecock Bay have problems. So this is a regionally significant habitat, home to the Shinnecock Nation. I want to recognize a couple of members of the Shinnecock Nation who are here tonight, who are working with us on an Indigenous Advisory Council, and generations of baymen who plied the waters here. So what were some of the problems? This is what the water looked like when I first got here. Looks kind of like the color of my cup of coffee, brown. And this had been going on for decades. For decades, we had annually occurring brown tides, and they really are brown. And other harmful algal blooms, including red tides. Those of you who have heard of red tides, you know that that can cause something called paralytic shellfish poisoning, and that can be fatal to humans. So we not only had something that was unsightly going on here and something that was affecting the life in the water, we had issues that were really serious in terms of human health. Big problems. Because the water was brown, sunlight couldn't penetrate through, the seagrasses all died. Just like any other plant, if you don't have light, you're not going to grow. The clam population collapsed for a variety of reasons. The water quality was bad. Also, there weren't enough regulations and too many clams were taken out of the water. So we had a really degraded ecosystem. And I would say that Shinnecock Bay was in a death spiral. So my colleagues had done a great job of documenting the problems in Shinnecock Bay. A lot of them have been here way longer than I have. And, I asked, well, what would you think about trying to fix the problem? And at first they looked at me like I was nuts. Like, fix the problem? Because we're scientists, we study things, we document things, we try to understand why, but fixing problems is another thing. Also, it seemed like the problems were so big and so difficult, that it was a really risky thing to take on. But we all thought about it, got back together, talked about it, and decided, yes, let's do it. Let's try to do it. And so something called the Shinnecock Bay Restoration Program was born, and it really got going in about 2010, 2011. We started doing some pilot work. And in 2012, we got major funding to really start doing this in earnest. And I'd like to recognize my co-principal investigators. Same people have been working on this project since the beginning. So how did we decide to tackle trying to improve the water quality in the bay and stop these harmful algal blooms? We didn't have a lot of money. We got some money, but not a lot of money. Things like building sewage treatment facilities were just out of the question and very long-term. We had an immediate crisis we wanted to fix. So we went for a nature-based solution. And what we did was decide to buy large hard clams and to put these clams in Shinnecock Bay to restore the natural filtration capacity of the bay. And the Southampton town trustees, who make decisions about the bay bottom, agreed that if we created these areas, they would close them to fishing. And they would patrol the waters and make sure that these were really sanctuaries for the clams. Now, we calculated that we needed about 33 million clams to eliminate harmful algal blooms in Shinnecock Bay. At the time, clams were about $1 a piece. Where were we going to get $33 million from? We weren't going to get $33 million, but we could get enough to plant a significant number of clams. And what we thought is, not only would these clams not get fished out, but they would grow, reproduce, may have offspring, their offspring would have offspring, and over time, we would get to that number of 33 million clams. So I'll tell you now, we exceeded that number. Took a long time, but it eventually happened. So what the idea was, we were going to plant clams. Being a gardener, I actually thought we were going to go out with shovels and take the clams and plant them. And I had no idea that really what it meant is that you throw the clams overboard in the water and they plant themselves. They do all the work. They burrow into the substrate and begin filtering the water. In addition to the hard clam sanctuaries, which was really our major push, we also built oyster reefs and reseeded eelgrass. So these were the interventions that were done directly, inputs into the bay that wouldn't have happened otherwise. After about 10 years, so a long time, we started to see signs of positive signs before that, but after about 10 years, the brown tides have been completely eliminated. We haven't seen a brown tide, and I think now it's been more than like 8 years. Other harmful algal blooms have been greatly reduced. The clam population has flourished, not only in the sanctuaries where no fishing is allowed, but outside those sanctuaries, and the clam fishery that operates outside of those sanctuaries is doing really, really well. The economic return is amazing, in addition to the ecological return. The seagrass has made a major comeback, and now people can enjoy a safe and healthy environment. So whereas Shinnecock Bay used to look like this on a typical day, it now looks like this. We turned brown tide blue. In 2022, Mission Blue, which is a global organization, designated Shinnecock Bay as a global hope spot. This is, the idea that we can turn a system that is so degraded around is really cause for hope. And it was recognized by Mission Blue. It was not only recognized locally, we got the front page of the Southampton Press, but also at a meeting I spoke at the United Nations, UN leaders said that Shinnecock Bay is a global model for conservation. So this little bay is not only the first hope spot in New York, but it's really something that people globally look at as an example, a rare example where we were able to turn a very bad situation around. That's the beginning of my story. And a lot of this has been documented in the scientific literature. There's a lot of information on the restoration. Here's the official ribbon cutting ceremony that we had in 2023. Sylvia Earle, who is the head of Mission Blue and probably the most well-known, famous marine biologist in the world, personally came to cut the ribbon and to celebrate. So we talked about clams, we've talked about oysters, we talked about eelgrass and harmful algal blooms. What about the fish? The fish are much harder. We weren't going to add fish to the water. We weren't going to build a hatchery and put little fish in. It just wasn't something that we were even thinking of doing. So the question's always been, what was the fish population like when all this got started, and what is it like today? Has the restoration, which has benefited Shinnecock Bay and all the people who depend upon it, has it had any impact on the fish? And that's been really the focus of me and my team. And I'd like to acknowledge Konstantin Runtos, who's one of my former graduate students. He was there at the very beginning when we started doing these fishery surveys. Great to see you again, Konstantin. Okay, so how did we figure out how many fish and what kinds are here? We started out by using a method called bottom trawling. And this is where a net is put over the side of the boat. It's hauled along the bottom. We bring the net up onto the deck of the vessel. And then we sort the catch and count how many fish of each species we see. We measure a lot of them. We treat them very carefully, and we put them back in the water and believe that most of them do survive. We also tried something called baited remote underwater video cameras, where we would put bait out and video cameras, and the fish that we would see, we would record how many fish we saw. That's another way of assessing how many fish are in Shinnecock Bay and what kind. And then something called eDNA sampling that I'm going to spend a lot of time on began in the year 2020 and continues today. So what is eDNA? Most of you are familiar with DNA. Heard about DNA. The E in eDNA stands for the environment. In the case of Shinnecock Bay or other water bodies, what it refers to is that in the water are DNA fragments of all the species that live in the water. And it's now possible to take a water sample from Shinnecock Bay and to filter the water, to process it, and eventually, I say eventually because there are several steps involved, figure out how many fish of which kinds are in Shinnecock Bay. It's a non-invasive, non-destructive method, and you can apply it in very sensitive environments like eelgrass beds, like hard clam sanctuaries. We didn't want to be bottom trawling in a hard clam sanctuary that we worked so hard to build, or an oyster reef. And I wrote about this method in Science in 2018, and I got so excited about it that I said, you know, why aren't we doing this in Shinnecock Bay? And so we started doing it. So here are the two main questions I'm going to spend the rest of the lecture talking about. One is what species occur in Shinnecock Bay? What lies beneath? Because you can't tell by standing at the surface and just looking at the water. And also this question of, can we conclude that this restoration that's been so successful in so many other ways, has it also improved the fish and invertebrate community of Shinnecock Bay? So this may be something that isn't a surprise to anyone in the room, but it was a surprise to me. Back in 2008, I didn't know there were tropical fish that showed up in Shinnecock Bay. But every year we have tropical species that show up, usually in the late summer, early fall. They're carried up from the tropics by the Gulf Stream. And a lot of people do go snorkeling, scuba diving to enjoy watching tropical fish swim around. Now look at that seahorse. I always thought seahorses were tropical species, and most of them are. But that particular one is a native species of seahorse that lives right here in Shinnecock Bay. And it's a temperate water seahorse. I didn't know that. You know, I was surprised by all of these things. The picture in the upper right, that's a coral. Coral in Shinnecock Bay? Don't corals only exist in hot tropical environments? No, there's corals, stony corals that exist in Shinnecock Bay. Sharks. Did you know that there are sharks in Shinnecock Bay? I hope you're going to be able to sleep tonight because you're going to see a few more sharks as the day goes by. But there's an example of one of the common sharks we see here, the smooth dogfish. And there, that funny looking fish that looks like it's on its side, that's a summer flounder. You've probably seen this if you've gone fishing or watched other people go fishing, or if you go to the fish market and you're looking for fish. That's something that you'll see pretty often. It's delicious tasting fish. It's ecologically very important. It's also recreationally important and commercially important. Okay, so this is just for the trawl series to show you. It's kind of, once you get it, you'll get it. So this is for all the species we've seen. So we've seen 113 different species have come up in the trawl over the years that we've been sampling, 113 different species. So if you look at the far left, you see that most of the species we've seen in Shinnecock Bay, we've only seen 1 year. So a lot of species show up, then we don't see them again. And then so this is a U-shaped curve, and it's an unusual curve. Most curves that you see in biology are kind of bell-shaped curve, the normal curve. It's the opposite here in Shinnecock Bay. We see species either very rarely or very frequently. And you can see that there are a number of species we've seen every year for 12 years on the upper right. Same thing, this is just the fish. U-shaped curve. Most of the fish we've only seen once. And then there are a bunch of what we call the regulars that we see every single year. Who are these regular fish? This will show you a pictures and words, but these are the 12 fish species that we've seen every single year since we've been doing the trawl work. And there's some of the species I pointed out, the temperate seahorse. We also have American eel, summer flounder, winter flounder, pipefish, weakfish, anchovy, etc. Even though we see all those species every year, they vary dramatically in terms of which are the most abundant and which are the least abundant. So if you look at the line seahorse, it's on the bottom, we see very few individuals, even though we see that species every year. It's still rare. It's a rare species, but we do see it every year. Whereas bay anchovy, scup, Atlantic silverside, northern pipefish, these are fish that we see in much greater abundance. There are 7 species of invertebrates that we've seen every single year. And, blue crab, people tell me stories, my colleagues tell me stories that when they started working here, there weren't any blue crabs in Shinnecock Bay. They were a southern species. Then they saw them, but they didn't survive the winter. Then they started surviving the winter and reproducing, and now we see blue crab all the time. So things really change over time, but blue crab we now see all the time. And several other species of crab, shrimp, squid. Blue crab, which wasn't even here not too long ago, is now the most commonly observed invertebrate in Shinnecock Bay. Interesting how things can change. Okay, so I've given you a sense of what species are here. Now let's look at, has the restoration had any impact on the fish? So I need to define for you what a forage fish is, or a forage species. These are species that feed very, very low in the food web. So they either eat plankton for the most part. They tend to be small bodied, have a short lifespan, school. And we hypothesized that because of their short lifespan, if any kind of species was going to show a response to the restoration, it would be forage fish. We've got several different species. So in the graph on the left, you see that we did start to see an increase in forage species, statistically significant, whereas all the other species just flat lines, no real change. That was several years back. But now we're seeing a new chapter unfold with the fish in Shinnecock Bay. Some of the higher trophic level species that eat the forage fish are making a big comeback. And we see here black sea bass, summer flounder. Summer flounder have that big mouth with the big teeth. They're major predators in the system. They're starting to show a comeback. Winter flounder is kind of an unusual story. It's going up and down. It's a predator of some species, but for example, it's prey for summer flounder. So now we've got a new picture emerging where the restoration is moving up. Why did we think forage fish would be first? Again, because they're short-lived and they, you'd have several generations in a short period of time. The fish that live a longer time would take longer to respond because they have a longer life history. So now the forage fish curve looks like this. It shows that early in our time series, the forage fish were increasing, and now they're decreasing. There's still plenty of them, but where our hypothesis is that this increase in predatory fish, those predatory fish are feeding on the forage fish, and that is the reason why they're coming down now. So as time goes along, we're going to start to see big changes in the ecosystem as a whole. I know this is a really complicated looking figure. The idea, though, is for you to look. So we're going from left to right, the early years, the most recent years, at how many different colors stack up over time. And that's an indication of how many different species are contributing to the biodiversity in Shinnecock Bay. So you see, like in the third or fourth year, big swaths of blue. That means that we had an ecosystem that was dominated by these forage fish. And as time goes by, we're getting an ecosystem that is much richer, much more diverse, very different picture as time goes on. So the ecosystem's changing. And we're in the process of not only documenting what species are here, but how do they interact with one another? And how can we use a model of the ecosystem to predict what might happen in the future? Okay, so eDNA. This is sometimes how we collect water samples for eDNA. Doesn't look very safe, does it? But at the beginning, and for a long time, actually, for 5 years, we did what we call manual sampling. And somewhere or another, we would get something that could hold the water over the side of the boat. We sterilized it ahead of time, no contamination. We would put it on ice, rush it to the lab, and then we would filter the water to get the DNA stuck on a filter. And then we would do something that would actually get that DNA off. It's called extracting the DNA. Then sequence the DNA, eventually compare the sequences of genetic code with reference libraries on species that correspond to that genetic code so we can match them up, and then eventually come up with what species of fish and invertebrates are here. So I'm just going to show a few again, like there's a lot to talk about. I don't have all night, and neither do you. So I'm just going to give you some of the highlights. So one thing that we didn't necessarily expect, but is borne out, is that eDNA is much better at detecting a broader diversity of species. So this shows the 4 species of elasmobranchs. Elasmobranchs are sharks, skates, and rays that we found after doing trawling for something like about 12 years. I think I had that right. 4 species of elasmobranchs. When we started doing eDNA after the first year, we found 12 species of elasmobranchs. So we tripled the number of species that we knew inhabit Shinnecock Bay of this kind of elasmobranchs just by moving to a new methodology. Why is eDNA able to do this? And why is it superior in that regard compared to a net? But with a net, you have a net that has holes in it. So really tiny things can go through the net and not get retained. And really big, fast swimming things never go in the net. Whereas eDNA can detect the DNA of any creature, whether it's tiny, huge, fast swimming, slow swimming. So it's a methodology that can find many, many more species. And this is a good example for the elasmobranch group. We also found that in our restoration areas where we can't and wouldn't want to go trawling, the hard clam sanctuaries, the oyster reefs, we found that eDNA detected 5 species that were unique only to those areas. So when we compare eDNA versus trawl for the 5 years that we've been doing both, we can conclude that we detected a lot more fish species than trawls do. We can use eDNA in sensitive habitats that trawls cannot. The detection of these unique fish species in restoration sites is further evidence that the restoration is increasing the biodiversity of Shinnecock Bay. We've created these new habitats and they are attracting new species. eDNA is non-invasive, non-destructive, neither of species or of habitats. It can be deployed in a manner with a much smaller carbon footprint than trawling. And we used primers that are focused on detecting fish, because that's what the trawl worked on in our manual sampling. But when we use other primers, they're called primers that can look at other forms of life, we find even greater biodiversity. Okay, so what are we doing now? Instead of hanging over the side of the boat, we decided to try something that had never been tried before. And this was our 2024 fall expedition. And what we did, this is something that I had thought about for a long time. Is there a way that we could collect this eDNA information autonomously in a way that it could be used anywhere? And I met the person who is the CEO of the company that makes the device on the left. It's called the USV, Uncrewed Surface Vehicle. And you can see that big black on the top. Those are solar panels. So this is a solar paneled USV. The hoop makes it self-righting. So if it flips over, it rights itself. That USV can withstand hurricane force winds. And, it's just an amazing piece of equipment. But what it didn't do was collect water samples for eDNA analysis. So, we had the idea of trying to find an eDNA sampler that would be autonomous and to integrate these two technologies together so that we could have a way of collecting eDNA information in situ autonomously in a way that has basically zero carbon footprint and could be used anywhere. And we decided to use it first in Shinnecock Bay. Why? Because we know this bay, and we figured that we would be able to compare whatever we find using this system to what we've already seen with other methods that I've talked about. So there's the Data Explorer, and the machine on the right is the autonomous eDNA sampler. It's called the Roxy. And of course, we had to find engineers who would put these together. It had to be tested. This was a process to get this system together. And here it is. We started to deploy it in September of 2024. Sunshine Gums, who is sitting in the room, is in the front. I'm in the back. I'm showing this picture for a variety of reasons. So this sits on a trailer, and you just wheel the trailer down a ramp. And then when you have enough water, you just let it go into the water. It only takes 2 people to deploy this system. Compare that with what we've been doing with bottom trawling. We have a boat, you need a captain, a mate. We have a crew of usually 10 people on the boat to haul in the net, sort the fish, count the fish, identify the fish, measure the fish. So with only 2 people, we were able to deploy and retrieve this Data Explorer system. This is what it looks like from a water's eye view once it's out on the water. What you see in the front, the white, funny looking thing, is the how we use the eDNA sampler, how we hooked it up. We went through a big process also deciding where should we put it? Should we put it on the top? Should we put it on the bottom? Where would it make the most sense to integrate these technologies, and decided on the top. And after having done this expedition, we definitely feel like we made the right choice, because when this comes back to shore on its own, then it's much easier to handle the cartridges. There are these cartridges that get used, and each cartridge stops, opens up, takes a water sample, closes, filters the water, it moves out of position, and the next one moves into position. 24 cartridges at a time. So you can take 24 samples. There is a model that can do 48 samples. And probably you could do as many samples as you want eventually. So there you have it. So in basically less than a 2-week expedition, we got 100 eDNA samples. We get about that many samples in an entire field season using the manual sampling technique. The system that we were using is not only autonomous, but it can operate day and night, and it can just keep going and going and going. We sampled at 27 stations, including 9 we'd never sampled at before. And we got samples day and night across multiple habitats. The samples were preserved in situ and ready for sequencing as soon as they got back to shore. And we got really good spatial resolution. So let me give you an idea of what it's like to use this. There's a cloud-based system. You could sit at home with your computer and log into the portal and plot a course for this, I'll call it the DX, or the Data Explorer. And those circles are the plotted course. The ones with the bullseye are the places where we wanted it to stop and take a sample. So this is an example of one of the actual courses that we ran during the expedition. Now, even though this could operate entirely on its own, for this expedition, we made use of, we stayed up at night, or we had shifts basically, because 4 hours is about as much time as anybody can be focused on what's going on. But the Data Explorer has 4 cameras, one that looks forward, a port camera, a starboard camera, and then a thermal front zoom camera that works at night. So we could sit, again, you could be at home, you could be here, you could be at Starbucks, looking at your computer, and you can see what those cameras are seeing. This is just to show that at the same time that the Data Explorer is collecting the DNA samples for us, it's also measuring lots of other things. So examples are temperature, latitude, depth, salinity, chlorophyll, turbidity. All these environmental variables are continuously being sampled and recorded by the USV. So we have an incredible depth of information on the environmental conditions, which again is very different from what we're able to do in a normal year, where we go out, we go out for the day, We collect our environmental data at each site, move on to the next site, collect data there. This is continuous and it's going on night and day. And the little red dots just show that the way this is programmed is that the USV goes into something called loiter mode. So it tries to maintain station while it's taking the sample. And it allows a little bit of drifting to occur, but if the drifting goes too far, it brings it back to where you want it to be. This is what our sampling plan was. We wanted to do day and night sampling. We did some trawling the same day that we used this so that we can compare on the same day what the trawl saw versus what was seen in the eDNA. We wanted to look at the Eastern Bay separate, pretty comprehensively, in some of the runs and the Western Bay. So there's lots more that we could talk about, but I'm going to wrap this up pretty soon. At this point, I think the main message here is that we showed that this works. This system works. It's a great way to get eDNA samples and to do it in a way that is high resolution. Some of the things that we were able to find were things like differences between the night and the day. So I'll just give you one example. We found the new species of shark during the expedition. It's a mako shark. So for those of you who are afraid of mako sharks, maybe don't go swimming at night in Shinnecock Bay. But we only saw that shark at night, not during the day. And so we were able to look at day-night differences. We saw some big differences in biodiversity in different habitats. And we had such great spatial resolution that it enabled us to see a lot more. The detection of environmental events in real time. We actually had an observation of a kind of an algal bloom, not a harmful algal bloom, but we were able to watch it occur and morph over time. So here we are again with this crazy looking graph. I just wanted to show you for the last 5 years, comparing the trawl versus the eDNA. They both are showing the same pattern as we move forward in time. That Shinnecock Bay, the fish community in Shinnecock Bay is changing, and it's changing in a way that most scientists would say is a healthier bay, one that has a much richer biodiversity, not where just a few species dominate, but where you've got a wide variety of species and they're all better represented. Okay, so I'm about ready to wrap up. I think I've shown the evidence, at least I convinced myself, that yes, the restoration, which dealt with clams, oysters, and eelgrass, has had an impact on the fish in Shinnecock Bay. We've transitioned over the period of time from what I would say is a very depauperate state to a forage fish dominated system, to a more balanced system with broader trophic structure and greater abundance of recreationally and commercially important species. And we've shown, not just with the expedition, but also for the past 5 years, that eDNA not only has advantages in terms of being non-invasive and non-destructive, but it also is better at detecting a much broader spectrum of aquatic life and can be used in more habitats than trawling. And we've demonstrated that eDNA research can be conducted around the clock, autonomously, with a solar-powered USV, with the potential to use it in remote areas of the globe where fueling is really not an option. And there are lots of parts of the world where if you wanted to study what's going on, you really couldn't count on being able to get reliable fuel supplies. this does make it possible to do studies like this on the high seas or in really remote areas. And that's it. Thank you, everyone. All right. So are there any questions? There you go.
Alan, as I've been involved in the physical work on this bay, schlepping clams, bags of clams. How is the effect of what was done mostly in the western part of the Shinnecock Bay and happened in the eastern part? Is the eastern part really come up at the same time, or is it a slower process?
Well, so the restoration, so what he's saying is that most of these clams are put in the Western Bay. That was because the Western Bay was really in much worse shape than the Eastern Bay. But yeah, the answer is yes. Like, for example, the clam population. The clam population has skyrocketed. We started with maybe a few million clams. We got to 33 million and we just ran right past it. And now there are so many clams in the eastern part of the bay where people can go fishing, that it's a huge success story. So there was less of a water quality issue to be dealt with, but you see the results in terms of more clams, more fish, things like that. Sure.
I'm just amazed. I've never heard about the eDNA. It's absolutely incredible.
Yeah, it hasn't been very long that we've had the option of using a method like this, and now it's just starting to catch on more and more. In a lot of ways, I think our team we're one of the pioneering teams, in terms of having this much data at this stage. Yes.
I fish on the bay and, recreational, but the inlet or anywhere. But the sea robins kind of just take over.
Mhm.
It's almost impossible to really go flounder fishing because they just get it. It's just instantaneous. Like you go out there for an hour, you get 100 sea robins trying to catch anything. Is there differences in seasons? Are you seeing a change? And, is that decreasing from a couple years ago?
Are you talking about is the sea robin
Yeah.
population decreasing? I don't think we've looked at the data to determine whether or not that's true. Maybe someone else on my team has an opinion though. Libby, do you? Yeah, I mean, it's all over the place. Yeah, it's Sea Robin City. But I mean, it's a good question. So, I said we found 113 different species just in the trawl alone. If you look at the eDNA, forget it, from the expedition, because we not only used, we used another primer that can detect even more kinds of species, lots and lots and lots of species. It's a gold mine of a database. And a lot of the questions that you might want the answers to, we haven't asked yet. We haven't analyzed the data that way. But, it's something that we could look at. Christine.
I'll just point out that when we used to do the underwater video camera, they were baited. And we saw a lot of sea robins in the underwater videos using the bait. The trawl is a different method. It's less targeted. It's based on a GPS location of a standardized survey. So just anecdotally, the sea robins would show up in the video survey more than the trawl survey. I don't know about the eDNA.
Yes.
A lot of people talk about water temperature change, maybe not in such a short amount of time since you've been doing it, but have you noticed anything with that?
Yeah, so, we were really worried that all this great success would be thwarted by the fact that the temperatures are warming. And So far, Shinnecock Bay is holding up very, very well, despite the high temperatures. There are parts of the bay that we've seen that become deoxygenated during periods of intense high temperature. But those haven't lasted long enough to really make a big impact. But it's something that we're going to keep looking at. And then the modeling that I was saying that we're going to be doing, one of my students, actually a student and a postdoc who are sitting in the back, Yifan and Ja, who are putting together these ecosystem models, will be looking at how temperature interacts with the food web and with the species as a whole. And part of why we're doing this is to see, can we predict, what can we predict about the way the system will respond as temperatures, if they do, continue to increase? Yeah.
What's so also different about temperatures down south where the water is 85 degrees, 80 degrees, and it's clear? Is it related to nitrogen in our area? Is it related to other factors that are?
Yeah, so, well, we have a lot of reasons why the water in Shinnecock Bay is not that clear. One is that it's very shallow. And we've got kind of a lot of the bottom is muddy. So anything that stirs up the bottom makes it more turbid, harder to see. That's not necessarily a sign of like poor health of the water. It's just, a shallow estuary like ours. That's what you're going to see. But we also we have a tremendous amount of nitrogen coming in to Shinnecock Bay. And that's what was fueling these harmful algal blooms. Algae love nitrogen. And you put more nitrogen in the water, you get big blooms. So in general, we just have a lot more of what some people would call pollutants and others would call nutrients in the water in Shinnecock Bay. We have a very developed landscape, and we've got a lot of stuff going into the water. Shanae.
Yes, I was actually gonna add something to that area about the diversity of development right around the Shinnecock Bay. There's, certain areas that have never been touched, right, by anyone. And the DNA there versus areas where there's bulkheads or where there's more development and natural habitat has been gone. What do you see in comparison to the two as far as the DNA in the water and these different areas?
Great question. We'll have to analyze our data more. Yeah, and it's also, it's also another reason why doing the kind of work that we did with the USV is a great thing to do, because you can sample so much faster, so much more area, and you can look at things on a fine scale. So we could be going to places that are bulkheaded and places that aren't bulkheaded and comparing them. Yeah.
Has the seagrass increased the cover?
It has. So we just had a meeting last week and we looked at what the seagrass looked like when we started the project and what it looks like today. There is definitely a noticeable increase in the amount of eelgrass in Shinnecock Bay, and that's twofold reasons. One is that we collect seeds and spread those seeds around in the bay, so we're providing more seeds to grow, but it's also that the water quality is better, and so it's a better environment for eelgrass to grow. Even though there's a lot more eelgrass now than there was maybe 10, 12 years ago, there's still not a ton of it. I was amazed at how much area that could theoretically be covered by eelgrass is not. So I think we have more work to do there. Yes.
After your 2-week excursion in the water,
Mhm.
with your material back, how long does it take you to do your tests? And I know it could be 1 week, it could be a month, it could be whatever, but until you send it back out again to get some more. How long was the intervals between, because you can't keep collecting it and then not have enough time to analyze, right?
Well, so the beauty of it is that each one of these cartridges that opens and takes the water sample, it actually squirts a preservative in at the same time. And so the filter that results from that can last at room temperature for 2 to 6 months. So we've got plenty of time to keep taking samples. Because of that preservative, we can just keep taking samples and not worry about having to attend to the samples we've already taken right away. So that means you can do a lot more. Now, what takes time are the steps like extracting the DNA and then doing the sequencing. We don't have the facilities at Stony Brook to actually do the sequencing. What is the DNA sequence that comes out? So we have to send those samples out to a laboratory that can do that. And it's not that it takes the laboratory that long, but there's so much demand now for this kind of information that, it often will take, well, we actually we paid extra to get a rush job on sequencing last year. Because we were so anxious to see our results. But, you're talking about months usually from the time that you send the samples in until you get that back. And then what you get back is so much data. It's just like one sequence after another. And you have to match that up against known sequences of known species in order to come up with the kind of information I'm showing. So that's a whole other step that we call bioinformatics. Now, we do the bioinformatics in our group, but it is very time consuming. Yeah, there are just a lot of steps involved in this.
And get KI involved.
Yeah, right.
Do it faster.
I think we will be able to do it faster eventually. I mean, I wouldn't be surprised. The technology is moving so quickly, and the interest in this, the demand for it, the demand for more information about what do we have in the ocean, not just Shinnecock Bay, but what is out there in the ocean is just incredible. And I wouldn't be surprised if like 5 or 10 years from now, the sequencing was being done in situ out in the ocean and the results were being transmitted to us by satellite. We're not there now. We're several steps away. What we did, I think, is one big step forward. The fact that you can use a solar powered autonomous system to collect this information, that's a big step forward, but there are lots more steps that we hope can happen, and hopefully quickly. So, like, I don't know how many of you follow ocean policy. Christine mentioned in introducing me about the 10 by 20 initiative. So in the year 2015, the United Nations agreed to the Sustainable Development Goals. And there was, for the first time in history, a goal for the ocean. And that goal was to try to protect 10% of the ocean by the year 2020. We worked hard on a campaign. At the UN, we would bring in scientists to talk with dialogues with ambassadors, to talk about what marine protected areas were, why they were important, what they could do for us. And during the time that we worked, from about 2016 to 2019, the amount of area protected quadrupled from just about 2% to 8%. It was really pretty amazing, the amount of progress. What happened in 2019, 2020? COVID. And priorities changed. Countries' priorities changed from trying to protect areas of the ocean to trying to keep people alive from dying of COVID-19. And, that's the kind of thing that happens in the world. So for a while, we were making great progress, and then things have stalled at about the same level since then. Despite that, a couple of years ago, there was an agreement reached to protect 30% of the ocean by the year 2030. So now our ambition is even higher. How are we going to do that? How are we going to decide first of all, what's worth saving? What should be the highest priority areas? The work on eDNA that, we're showing and showcasing here could be a really crucial element of that. Because with a remote operated system, we could be sampling out areas in the high seas that we haven't been able to sample before. And we could say, hey, this has a lot of biodiversity. This doesn't have so much biodiversity. This is an area that seems stable. This is not. So there are these really big things going on. The 30 by 30 commitment. And then there's something called the High Seas Treaty. How many of you have heard about the High Seas Treaty? Okay, so a bunch of you have. So that was agreed to in principle. When was it? Just like a year and a half ago? A year and a half ago. After 20 years of negotiation at the United Nations, because things have to be agreed by consensus at the United Nations. And consensus of difficult subjects can take a really long time. And in this case, it took 20 years for there to be an agreement that we should have a treaty that governs what happens out of areas within national jurisdiction, the high seas. So even though that's been agreed in principle, there's still a lot of work to do. One is that will not come into force until 60 countries ratify the treaty. So far, we have 20 countries that have ratified the treaty. People are optimistic that we might get a lot more. The UN Ocean Conference is going to happen in France in about a month. And we're hoping to see a lot of announcements by countries that they're ratifying the treaty. Even then, the nuts and bolts of the treaty have to be worked out. So we have this idea that we're going to manage the high seas. Well, how? How are we going to create the scientific bodies, the management bodies, the enforcement bodies that are going to do the work? And what principles will they use? And how will they be selected? And what will be the qualifications? Those are things that we're working on now. Okay. I think I've tired you out. Thank you very much.